Hi biologists, let's start by looking at the learning objectives for this lesson. At the end of this lesson and following the biology syllabus, you should be able to explain tissue culture and refer to any two applications. What does this actually mean? What are we trying to understand? Well, you have to be able to explain tissue culture and you have to be able to state two ways in which it can be used. Let's look at tissue culture and make a start by looking at the definition. Tissue culture is the growth of tissues in or on an artificial medium in a petri dish outside an organism. We met this idea of in vitro growth before. In vitro growth, the growth of cells or tissues outside the body in an artificial environment, in glass. Remember, in vitro is the Latin word for in glass, and we met this concept when we looked at the section on IVF treatment in human reproduction. Now, tissue culture is a technique in which individual cells are grown and divide in a bath or petri dish or test tube of sterile, no microorganisms present, nutritive fluid, which often contains hormones and growth substances. The hormones stimulate growth and glucose acts as a source of energy. The fluid might also contain other substances necessary for growth like vitamins and minerals. Now, tissue culture does have a number of limitations. First of all, the growth medium must be kept sterile. In other words, there can be no microorganisms present. Bacteria and fungi grow fast. They make wastes that contaminate the container and kill the growing cells. Now, an objective of this lesson is to be able to explain two applications or two uses of tissue culture. The first application or use of tissue culture is micropropagation. This is really asexual reproduction in plants. We go into this section in a bit of detail here because this section will make a reappearance when we are studying asexual reproduction in plants at a later stage. Now, micropropagation is the growth of a large number of plants from very small plant pieces or cells. We can see the word propagation here, meaning growth or reproducing, and we can see the word micro, meaning very small. Hence, micropropagation or the growth of plants from very small plant pieces or cells which are microscopic. The second application or use of tissue culture is in the use of skin grafts. This is where human skin can be cultured or grown and given to badly burned patients in order to heal their burns. So we will look at each application in turn. Micropropagation, or sometimes called plant breeding, is where a plant is cut into many small pieces. The cells or small pieces are grown in vitro, don't forget, in glass, perhaps in a petri dish, on a suitable medium. The medium, remember, has to contain growth hormones and glucose as a source of energy. After some time, a clump of similar cells called a callus is formed. The sound of this sentence helps me remember. A clump, see, similar, cells, called a callus, is formed. We might have such alliteration, is it, in English class? A clump of similar cells called a callus is formed. So that helps me remember this step. Having formed a clump of similar cells called a callus, the growing conditions are changed, so that the callus will grow into a young plantlet or small plant, rather like weaning a baby from drinking milk to solid food. The growing conditions are changed here, so that the cells grow into a young plantlet. When the plantlets are large enough, they can then be planted out like normal plants. As a reminder, the clump of similar cells called a callus grow by mitosis, so that's why all these cells are similar. The cells are actually clones of one another, because the definition of mitosis is that it is nuclear division producing two new cells, each identical, genetically identical to each other and to the parent cell. This is an important point to keep an eye on as we move through the section. So in summary, micropropagation is where a plant is cut into small pieces, grown in vitro to form a callus, grown perhaps in a petri dish. The cells divide by mitosis, the growing conditions are changed, the cells are still clones of each other, being genetically identical to each other as they were produced by mitosis, eventually forming a young plant, and when the plantlets are a little larger, they could then be planted out as normal. So, following this technique, a large number of plants can be grown. They're genetically identical to each other and to the parent plant and are clones of each other. Now, what are the advantages of micropropagation? Well, first of all, once the technique has been developed, it is a cheap way to produce a large number of similar plants in a short time. Now, as a gardener, we might want to have a cheap way of producing similar plants. If I'm interested in producing pink orchids, I will want to be sure of my colour, pink, and if I follow this technique, then I will be sure to produce a large number of such similar plants in a short time so that I can sell them quickly. Micropropagation is also not dependent on the seasons. It can be carried out at any time and done throughout the year. A small amount of initial plant tissue is needed. I just need a small clump of cells, and from one small clump of similar cells, I can produce a large number of plants in a short time. Another advantage is that at the end of this technique, you will have rooted plantlets, which saves grower time in that we can produce flowers quicker than waiting for the plants to grow from seed. The disadvantages of micropropagation are that there's no variation, so the plants are equally susceptible to disease and they could all die. We can understand this from our experience with COVID. Let's suppose everyone in the class catches COVID. Because there's variation in the class, some people will show no symptoms, some people will be coughing and have sore throats, others might wind up going to hospital, and some people might pass away as a result of the disease. Hopefully not. But you get the idea. Because there's variation in the classroom, different people will react to the disease differently. If there were no variation in the classroom, everyone would have the same immune system and everyone would be wiped out as a result of COVID. So here the idea is, because the plants are all clones of each other and there's no variation, their immune systems will be identical, they will be all equally susceptible to disease, and there is the possibility that they could all die from it. Another disadvantage of micropropagation is that it requires more labour and costs more money from that point of view. And not all plants can be tissue cultured because it can be difficult to find a growth medium for the different species of plants. Now, let's look at the second application of tissue culture, the use of tissue culture in the process of growing new skin for badly burnt patients. Human skin can now be cultured or grown to heal severe burns. In the past, a piece of healthy skin would have to be taken from elsewhere on the body and grafted on to the site of the burn. Nowadays, skin epidermis cells can be isolated from a small skin sample belonging to the person. The fat would be detached as it's not needed, and the skin cells are incubated or kept at a favourable temperature in an incubator or oven, and the cells cultured in an artificial medium. They are then grafted onto the burn as a unique layer of cells organised in a sheet. Bone marrow stem cells are often used to grow new skin as well. Stem cells have the potential to differentiate or grow into different tissues as needed. Now, the benefits of tissue cultured skin grafts are that there is faster healing and less scarring due to the burn. There is no need to remove skin from another part of the body, because where the skin is taken from can sometimes be sorer than the actual burn itself. And apart from that, we don't want to be taking skin from a healthy part of the body. Most importantly, the new skin is identical to the patient's skin, so the skin graft is not going to be rejected. And there you have it. Practice in a jotter. Now that we've reached the end of our lesson, have we achieved our objective? Can you explain tissue culture and refer to any two applications?